University. He works on a flat model. He will tell us the genetic platform for functional profiling and the visualization of sphingolipid metabolic network.
I'd like to thank the organizers for the opportunity to talk here. So I enjoy walking the beach, also the cultural heritage Chichen, which is amazing. So thank you very much. And today I'm gonna talk how we study sphingolipid in the fly brain using the Drosophila as a model. So the brain is a complex organ with a lot of lipids. So in fact, about more than 60% of the dry weight are lipids. And the imbalance of those lipids are associated with neurodegenerative diseases, including Alzheimer's, Parkinson's, etc., many diseases. And today I'm going to talk about sphingolipids. And sphingolipids are discovered in brain extract in the 1870s, so more than 140 years ago. And they are named after the mysterious sphinx because of their structure and also because of their enigmatic nature of their biochemistry. And over the past century, the biochemistry has become more and more clear. However, how they are regulated at the systemic level are still largely unknown. So shown here is a simplified pathway how different sphingolipid species are synthesized and degraded. And so if you take this ray pathway, it's the synthesis of ceramide, and different species of those sphingolipids have diverse functions, such that they serve as a membrane component, and also they regulate signal transduction, lipid storage, and in neurons they regulate myelination, especially the sphingomyelin. So today I'm gonna use the fly model to study the mechanism, and I'm gonna focus on two enzymes. One is the conversion of dihydroceramide to ceramide. That is the last step of the de novo synthesis of ceramide. And this step is regulated by the DEX1 enzyme, and mutation of this DEX1 enzyme are associated with leukodystrophy, which is a brain disorder affecting the white matter. And another enzyme that I'm going to introduce is the hydrolysis of sphingomyelin back to ceramide. And there are a class of sphingomyelinases, and SMPD4 is one of them. And recent identification of SMPD4 mutations associated with a disease called microcephaly, which is the small brain syndrome. So let me start with BEGS1. So in 2019, about 5 years ago, these mutations in DEX1 are identified to associate with, so it's a rare disease associated with leukodystrophy. And as I just told you that BEGS1 is the last step converting dihydroceramide to ceramide. And this is the de novo synthesis. And those are back-to-back paper show that the mutation in here causes a severe syndrome affecting the white matter, but the disease-causing mechanism at the time are still not clear. So let me tell you what we learned from the flies. So on the right, you see that sorry, on your left, my right, or this is the de novo synthesis of pathway in human as well, and this is in fly. As you can see, there's a lot of similarities, and the pathways conserved, and also the enzymes are conserved in humans. So that's one in flies called IFC, and we found in a genetic screen for neurodegeneration that loss of DEX1 IFC leads to the morphological degeneration of the sensory neuron photoreceptor, that is the fry eye, and also we did the functional assay and showed that loss of DEX1 IFC leads to functional degeneration also in photoreceptor, in that the photoreceptor response to light reduce their response to light. And those two defects can be rescued by taking back human transgenes, suggesting the functional conservation between human and fly. So we can use this fly model to study the function, the endogenous function of DEX1 IFC. And we also did the lipidomics, and we found that so this gene controls the conversion of dihydroceramide to ceramide. And lipidomics show that in the fry brain, when we knock out DGS1 IFC, it leads to the increase of dihydroceramide and also decrease of the product ceramide. And this is also the same case in the patient fibroblasts in the data group. And so another question comes to what kills the neuron? Is that because of too much precursor dihydroceramide or too little product ceramide? So to test this, to examine this question, we put back the product and also downstream lipids and found that there's no rescue effect. So on the other hand, when we inhibit the upstream enzyme, either using chemical inhibition or using genetic RNAi, we show that the defect, both the morphological defects as well as the functional defects, are reversed by those two approaches, meaning that it is because of too much, the neurodegeneration is due to neuronal accumulation of dihydroceramide. So in the interest of time, I skip many validating data, but also in the human cell line, we use a drug to inhibit DEX1 IFC. We saw the apoptotic marker, meaning there's a cell death being reduced upon dose of DEX1 IFC, and this can be reversed. When we inhibit the upstream enzyme using another drug. So double inhibition reverses the phenotype. And so this is the model. Loss of IFC leads to the accumulation of the neurotoxic dihydroceramide, and which leads to the oxidative stress, ROS accumulation that kills the neuron. And neuron activate. Neurons try to protect themselves by activating lipid clearance machinery called lipology, which I don't have the time to get into. But in this model, there are several manipulating nodes. Like we can inhibit the upstream enzyme, or we can use antioxidant to clean up the ROS to rescue the degeneration. But now, here comes the next question. So we observe the increase of oxidative stress. So what kills the neuron? Where is the loss from? In other words, how does loss of the lipid modifying enzyme leads to the genesis of oxidative stress? And usually, when it comes to oxidative stress, people think about mitochondria. So we checked where the ROS is from, and using two probes. One is the cycle RhoGLP, which detects the ROS in the cytoplasm. And here is a genetic trick. We make a tissue, the photoreceptor, half wild type and half mutant. The red dots indicate wild type, and the black indicate mutant. And in the mutant, in the IFC knockout background, we saw that the quench of RhoGLP, indicating there's a oxidative stress in the cytoplasm, and that is not the case when we use micro-RhoGLP, indicating mitochondria mitochondrial function is not affected in regards to ROS genesis. And we also did a genetic screen. So first we made a CRISPR knockin cell line of human neuronal cells with the DEX1 disease point mutation. And then we feed the cell, we treat the cell with different inhibitors of those cytoplasmic ROS generating compartments. And we found that apocynin reverse the genesis of the oxidative stress. And apocynin is a NADPH oxidase inhibitor, so it inhibits NAD's activity. And then we treat the IFC knockout fly with this apocynin, and we saw the reverse of the light sensing function. So indicating that the oxidative stress came from the NADPH oxidase in the cytoplasm. So now we know that apocynin inhibits NADs, and we also know from the literature that NADs activation requires REC1 membrane association. But we don't know that how does this lipid modifying enzyme regulate REC1 membrane association. So we hypothesized that REC1 regulates IFC DEX1 regulates REC1 for ROS genesis. And here's the hypothesis model that EGS1 converts dihydroceramide to ceramide in the wild type background, and REC1 in the wild type background associated with plasma membrane. However, in the DEX1 IFC mutant, when the sphingolipid composition is altered because of this the loss of this enzyme, it makes the it altered the association of REC1 to a membrane such that it alters the preference of this protein association. So REC1 would mislocalize to a compartment that activates the ROS genesis. And to test this hypothesis, we performed a liposome binding assay, which in which we manipulate the lipid composition to mimic different lipid compartment, including autophagosome and membrane wrap. And in this assay, we manipulate the content of the hydroceramide. And we saw in the middle that when we increase the dihydroceramide, we saw the association with the activated REC1 to autophagosome, but that is not the case in the membrane rat. Interestingly, we saw the decrease of REC1 to membrane rat, but it increased the association to autophagosome. And this is also the case in the patient fibroblast that we show that the REC1 from the patient fibroblast lost the association to the membrane graft on the right. So the summary is that upon loss of DEX1, it increases the content of dihydroceramide and alters the binding of active REC1 to membranes. And this leads to the mislocalization of REC1 to the endolysosomal compartment, which induces the REC1 NADS-dependent cytoplasmic ROS genesis. And this is probably an example of using fly to understand the mechanism of how a human disease associating lipid modifying enzyme, how it works. But now I wanna take this beyond the single mutation and look at the whole picture. Here again is the whole network of sphingolipid modifying enzyme, and those enzymes, several of them, if not all, associated with diseases upon mutated. So many of those diseases have those neuronal symptoms, including Parkinson's, epilepsy, leukodystrophy that I just told you, ALS, etc. So, but we all know that those diseases, they are different, indicating that these enzymes, they probably affect different cells. They do different things in different contexts. So if you want to study an enzyme, the endogenous function of the enzyme, it's better to study it in endogenous context. So we want to look at, so we have some goals. First, we want to profile the enzymes. So here's the goal. So the aim of this part of research is that first we want to profile the expression pattern of those enzymes, including their transcriptional expression and subcellular localization. And then we want to manipulate all of the enzymes by to study its in situ function. Meaning we want to knock out the protein at the right place at the right time. And then we want to visualize when we when we knock out those lipid modifying enzymes, how does the lipid be altered? Such that we are gonna make a sphingolipid biosensor to help us visualize the change in lipid landscape. And I'm not gonna bore you with those genetic tricks, but basically, the first step is to make the reporter flies of all the lipid modifying enzyme. That is about more than 50 genes in fly. So 50 reporter flies, and we are gonna do the systemic gene profiling, and then so the genetic trick is basically that we use CRISPR to knock in a genetic fragment at the endogenous loci, the sequence of the endogenous loci, to take the coding region with H8 and also put in a GAL4, that is an expression component controlled by the entire cis regulatory element. And then, because now this protein is tagged with H8, we want to knock out the H8 tag protein with a system called degrade H8. So the idea is just that the H8, we fuse the H8 nanobody with E3 ligase. So this E3 ligase will recognize the H8 tag protein, and so expression and also degradation would lead to some change in lipids. And we want to visualize the lipid. So we make some lipid biosensor, and this one case is that the delivery of sphingomyelin by a sensor called ODA. So ODA labels sphingomyelin, and this were done by a couple of many talented students in the lab. And now with those reported, we want to first study the brain, which is the complex organ, and several of those brain structures have already well-defined function. So we want to look where those genes are expressed, and on the right you see the heterogeneous expression pattern. They are not expressed ubiquitously. Some of the genes doesn't express even in the brain at all, but we know that they all express because we also check the gut. In gut, almost all the genes express, but that's not the case in the brain. So this suggests the heterogeneity, and also it suggests that still non-autonomous function, because when the enzyme is not there, it means that either the cell does not need the enzymatic function, or it has to get the product from other places. Those are still non-autonomous that. So then we superimpose those images to find which enzymes work together. And we found a set of enzymes, they localize to the same neuron, and those enzymes interestingly regulate the synthesis and degradation of the seroma. And this model we are still testing. So now the last part, I want to use this sphingomyelinases as an example of how we do those profiling work. And sphingomyelinases regulate the hydrolysis of sphingomyelin to seroma. And in fly, there are 5 sphingomyelinases. In human, there are 3. And those are sphingolipinases in fly. Three of the five expressed in the brain. Two of the three expressed in neuron, and for those two in the neuron, they have a distinct subcellular localization pattern. So I want to focus on this SMPD4 gene. It localized to the cortex neuron. Subcellularly, it is in the compartment of the nuclear envelope and ER. So we want to know what it does there. So we want to know because recently there's a paper showing that SMPD4 mutants cause a disease, the small brain syndrome, microcephaly. And so what we know from the systemic profiling is that SMPD4 expresses in specific brain region. And that region in fly is called mushroom body. That is the structure regulating the cognitive function. And subcellularly, it localizes to ER and nuclear envelope. And we also know that human mutation encodes for truncated protein, which likely represent those function variant. But what we don't know is if the knockout fly resembles the human microcephaly syndrome. So, and also does the knockout alter sphingolipid landscape? So we also want to know the mechanism. If the knockout causes sphingolipid imbalance, that leads to the morphological and functional effects resembling microcephaly. So this is what we have found. So to make a fly model, first we make a knockout fly of SMPD4, and this is the generation of the knockout fly. And then we use this in the middle, the ODA biosensor to check for the sphingomyelin level. And upon loss of SMPD4, we saw an increase of sphingomyelin at the very right there. And we also performed lipidomics to see that the ODA signal is consistent with the lipidomic data showing the increase of sphingomyelin. And it is not only the increase of sphingomyelin, we also show that the SMPD4 knockout increase a specific species of sphingomyelin, that's the C18 classes. So we speculate this C18 classes may be the specific classes responsible for small brain syndrome, microcephaly. And at the cellular level, loss of SMPD4 leads to the phenotype of nuclear brevity, that is the swelling of the nuclear envelope. And this is the same case as well as when we use degrade HA to do the protein knockout of SMPD4, similarly leads to the nuclear brevity phenotype. And this phenotype is rescued by human SMPD4, not shown here, so again suggesting the evolutionary conservation. And at individual level, this SMPD4 flag represent microcephaly-like syndrome. So in the profiling, we show that SMPD4 expresses in region regulating learning and memory. That is the flag cognitive center. And we first count the cell number in this region, and we show the reduced number of neurons in this region. And functionally, we perform a learning memory assay and show that loss of SMPD4 leads to the learning defect. So together, these morphological defects as well as functional defects suggesting that SMPD4 knockout represent microcephaly-like phenotype. So what I have shown you so far is that we have made a system to do spatial temporal profiling and the manipulation of sphingolipid network. And this, interestingly, we found the functional organization of this sphingolipid modifying enzymes in distinct brain regions and cell types. And using SMPD4 as an example, it regulates specific lipids for nuclear envelope integrity and brain functions. And this one-for-all system is not just just for disease modeling, but can also help us understand the sphingolipid biology in systems level, at least in the model organism, full-fledged Drosophila. And with that, I'd like to thank people in my lab, a couple of very talented students, Fei Yang, Tai Hong, Guan Shi, and also my long-time collaborator, Su Yiguang at the National Taiwan University Hospital, and also several other people working together at the Sphingolipid Frontier and the funding agency, and I'll start here with questions. Thank you.
So, did you have a profiling of the for expression level of SMP before you, right? Which cell type express more this SMP?
So it expresses more in neuron. So we have done the comparison of neuron and glia. So the reason we didn't do deep cell type specific expression is that at the first step we want to know where they express, and then we have those for to knock out the neurons that area. But the specific neuronal identity we don't know.
Actually, Shichang, I do not quite understand the red one part.
Okay.
Why red one needs to be in the autophagosome to reduce NOx? So NOx is effector, but when it is activated, then the effector comes, right?
Right.
Why? So on the plasma membrane, it is also activated. What's the difference between the?
So so there's some data that I skipped. First, we found that the red one is so we did those biochemical fractionation and found red one in plasma membrane as well as in the endosomal compartments. But from the literature, so we actually wanna make a liposome that represent the whole endolysosome. But in the literature, we cannot find this paper telling us what a standard endolysosome has, what kind of lipid composition. And this is the one paper that shows the autophagosome lipid composition. So we just use liposome binding acid to represent autophagosome, because we only know the lipid composition of autophagosome. And it would be nice if we know the composition of endosome so that we can mimic endosome.
So what do you mean is that the lipid component difference makes red activation difference, or?
That's our speculation.
Okay.
So because we really don't know, but the liposome binding acid tells us that they don't like membrane wrap when we increase the dihydroceramide content.
Okay.
So that's all we know. And also in the patient fibroblasts also show the same. The EGS1 supposed to be made more of dihydroceramide, and we purified the membrane, and we saw less red one association to the membrane. We don't know why, but that's what we see.
So it could be because of the lipid content and recruits different red regulator, right? And then makes a difference.
That's the working model.
Okay, understood. Okay, no more question. That's it.